Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. I'm going to present a medication that has been asked of me for quite some time. It's a medication that I am very interested in, but yet had not done my due diligence or a deep dive into until just very recently. And what I have discovered about this medication has been very exciting. And the medication that we're going to talk about is called ivermectin. Many of you are familiar with this medication. It is a well-known drug used for parasitic infections, both for humans and animals. And it has been demonized widely in the media when people have considered the medication for purposes outside of the initial or the original indication. And we saw that during the pandemic, and we're seeing it play out now. But the truth is that there is a ton of research on it being used as a repurposed medication, both for viral infections as well as for cancer. And given that this is Cancer as a Mitochondrial Metabolic Disease series, we're going to talk about its role in cancer over the next coming videos. The way we're going to look at ivermectin is going to be a very stepwise approach. We're going to look at ivermectin's effects on cancer stem cells and its effects on pro-growth signaling pathways today. Then in the next video, we're going to look at how ivermectin affects glucose metabolism, glutamine metabolism, induces oxidative stress, and activates apoptosis. And then in the last video, we're going to talk about its synergy with the standard of care treatments, as well as synergy with other metabolic inhibitors. And we're going to look at a couple of case reports of people with cancer who used ivermectin under the care of their physician and seeing what kind of results that they had with the medication. So without further ado, let's get into it. So the first pathway that was discussed when looking at some of these repurposed medications was the hypoxia-inducible factor pathway. And I think that's an amazing pathway to get the ball rolling when talking about repurposed medications, because it is so critical as a driver of the Warburg metabolism and enhanced glutaminolysis. So the first paper that I found was published in October of 2020, and it's titled Targeting Tumor Hypoxia and Mitochondrial Metabolism with Anti-Parasitic Drugs to Improve Radiation Responses in High-Grade Gliomas. And although this paper is not specific to ivermectin, it gets into the world of using medications off-label for purposes other than their intended approved purposes for something like cancer. And it says here that herein tumor hypoxia is identified as a significant contributor to the radioresistance of high-grade gliomas, as oxygenation is critical for the effectiveness of radiotherapy. Hypoxia plays a fundamental role in the aggressiveness and resistant phenotype of all solid tumors, including high-grade gliomas, by upregulating hypoxia-inducible factors, which stimulate vital enzymes responsible for cancer survival under hypoxic stress. Since current attempts to target tumor hypoxia focus on reducing oxygen demand of tumor cells by decreasing oxygen consumption rate, an attractive strategy to achieve this is by inhibiting mitochondrial oxidative phosphorylation, as it could decrease oxygen consumption rate and increase oxygenation, and could therefore improve the radiation response to high-grade gliomas. This approach would also help in eradicating the radio-resistant glioma stem cells, as these predominantly rely on mitochondrial metabolism for survival. Here, we highlight the potential for repurposing anti-parasitic drugs to abolish tumor hypoxia and induce apoptosis of glioma stem cells, Current literature provides compelling evidence for these drugs, atovaquone, ivermectin, proguanil, mefloquine, quinacrine, could be effective against cancers by mechanisms including inhibition of mitochondrial metabolism and tumor hypoxia and inducing DNA damage. Therefore, combining these drugs with radiotherapy could potentially enhance the radiosensitivity of high-grade gliomas. And it's hard not to chime in when reading this with this abstract because I do believe that there is little or no OXPHOS or oxidative phosphorylation going on in cancer cells due to dysfunctional mitochondria and mitochondria that are effectively switched off by PDK, which is even in their own graphic here. The oxygen consumption they believe is related to reactive oxygen species and not OXPHOS. And for evidence about that, we can refer to several other sources, including Seyfried's papers. But regardless, their thought about using these repurposed anti-parasitic medications is attractive. And in particular, they're talking about the effects by these medications on the hypoxia-inducible factor system. And as you can see here, and I've shown in other graphics, and I'll show in graphics in the coming videos, in particular, we're talking about glucose and glutamine, but hypoxia-inducible factors are responsible for every part of the Warburg metabolism and aerobic glycolysis, as well as enhanced glutaminolysis. So we can see here that hypoxia-inducible factors, number one, will upregulate PDK, pyruvate dehydrogenase kinase, which will ultimately shut down mitochondrial metabolism, making a strong point against what they're trying to say here about cancer using OXPHOS. Second, it will upregulate all of glycolysis. It should have another arrow here with glycolysis and all the glucose machinery. So that would include hexokinase, phosphofructokinase, all of the glycolytic enzymes, including pyruvate kinase, lactate dehydrogenase A, etc. It will also upregulate lactate transporters. It will upregulate glucose transporters, and it will help to deal with the excess oxidative stress that is being put on the system when you effectively block OXPHOS when oxygen is present. You're also going to see an uptick in glutamine transporters as well as glutamine enzymes that metabolize glutamine. And due to the enhanced glucose metabolism and glutaminolysis, we'll also see an uptick in lactic acid as well as succinic acid. So what they're proposing is that these anti-parasitic drugs, through a variety of mechanisms, because we're talking about multiple different drugs, not just one, but I use this paper to get us into the idea of anti-parasitic medications being used for cancer. It's saying that it's going to help block the cancer stem cells, mitochondrial function, increases in reactive oxygen species, and increases in apoptosis. And we have seen in prior videos where things like mebendazole and fenbendazole can have powerful multi-pathway and multi-targeted approaches to eradicating cancer. The same is going to be the case for ivermectin. So the first thing I wanted to touch on was ivermectin and its role in affecting the cancer stem cells. And this paper was published in February of 2018, and it's titled Ivermectin as an Inhibitor of Cancer Stem-like Cells. The aim of the present study was to demonstrate the ivermectin preferentially inhibited cancer stem cells in breast cancer cells and downregulated the expression of stemness genes. It says in the paper, ivermectin, a polycyclic lactone pesticide produced by Bacterium streptomyces avermitilis, is a broad-spectrum antiparasitic drug that has been used in human medicine since 1987. It is the drug of choice for treating onchocerciasis and strongyloidiasis, and a therapeutic option for mass population treatment campaigns for lymphatic filariasis. As a microfilaricide, a single dose of ivermectin is fast, effective, and well tolerated and causes little or no severe inflammatory responses. Commonly observed adverse effects from the treatment of ivermectin are mild and self-limiting and include skin reactions such as itching, musculoskeletal pain, fever, swelling of the face, joints and limbs, headaches and dizziness, lymphadenopathy, eye reactions, and pain from parasitic nodules. It later says that in the field of drug repurposing for cancer therapy, major efforts have been undertaken to identify drugs that may selectively or preferentially target the cancer stem cell population of tumors. The results of this present study demonstrate that ivermectin, an anti-parasitic drug approved for human use, preferentially targeted the cancer stem cell enriched subpopulation of breast cancer cell line MDA-MB231. I think this is important to realize because it's not well made clear in the media, but that ivermectin is an anti-parasitic medication that is approved for human use. It is not only for animals. Failure to successfully eradicate tumors is no longer amenable to local treatments is at least partly due to the existence of cancer stem cells, which are characterized by tumorigenic properties such as self-renewal, formation of differentiated prodigy, and development of resistance to therapy. In conclusion, results from the present study demonstrated that ivermectin preferentially targeted the cancer stem cell population and MDA-MB231 human breast cancer cells. Ivermectin has been demonstrated to be safe following treatment of millions of patients with onchocerciasis or other parasitic diseases, which makes it a strong candidate for further studies investigating its potential use as a repurposed medication or drug for cancer therapy. So I like this setting the tone that it is a strong and safe medication that is a strong candidate for the repurposed medication or drug for cancer therapy. I like that. Moving on. What I'm also going to show is that ivermectin has diverse effects on a variety of key signaling pathways that are responsible for the phenotype of cancer and prolonging or propagating the Warburg effect and enhanced glutaminolysis. The first pathway I want to discuss ivermectin having an effect on is the AKT mTOR pathway. And this paper was titled Ivermectin Augments the In Vitro and In Vivo Efficacy of Cisplatin in Epithelial Ovarian Cancer by Suppressing AKT mTOR Signaling. This paper was published in February of 2020. And it says here, mechanistically, ivermectin suppressed the phosphorylation of key molecules in the AKT mTOR signaling pathway in ovarian cancer cells. In addition, overexpression of constitutively active AKT restored ivermectin-induced inhibition of AKT mTOR growth and arrest. So what they did was they used ivermectin and it shut down this AKT mTOR pathway. And then what would happen is they would overexpress the gene and it would lose the effectiveness at growth arrest and apoptosis, which is showing that ivermectin, one of its major ways that it works is through this PIK3 AKT mTOR pathway. Our work suggests that ivermectin may be a useful addition to the treatment armamentarium for ovarian cancer, and that targeting AKT mTOR signaling is a therapeutic strategy to increase chemosensitivity in ovarian cancer. The next pathway we're going to look at is that of this YES pathway. And I'm going to show you guys the graphics that help us understand where these pathways are having an effect. So this paper is titled Anti-Tumor Effects of the Anti-Parasitic Agent Ivermectin via Inhibition of YES-Associated Protein 1 Expression in Gastric Cancer. This paper was published in November of 2017. And it says here that we found that MKN1 gastric cancer cells were most sensitive to ivermectin, whereas MKN7 cells were most resistant. In MKN1 xenografts, ivermectin suppressed tumor growth, and the sensitivity of MKN1 cells to ivermectin was decreased by YAP knockout protein. Ivermectin inhibited YAP1 nuclear expression, mRNA expression, therefore inhibiting nuclear accumulation of YAP1 in MKN1 cells. The survival analysis, low YAP1 mRNA expression was associated with better prognosis and 3 independent gastric cancer datasets. In conclusion, we identified ivermectin as a potential anti-cancer agent and found a promising novel therapeutic strategy for inhibition of gastric cancer progression by blocking YAP1 expression. The next pathway that is critical for cancer progression is this WNT beta-catenin pathway. And this paper was published in October of 2022 saying ivermectin inhibits tumor metastases by regulating the WNT beta-catenin integrin beta-1 FAC signaling pathway. It says mechanistically, ivermectin suppressed the expression of the migration-related proteins, inhibiting the activation of WNT beta-catenin integrin beta-1 FAC and the downstream signaling cascades. Our findings indicated that ivermectin was capable of suppressing tumor metastases, which provided the rationale on exploring the potential clinical application of ivermectin in the prevention and treatment of cancer metastases. Sounds pretty promising to me so far. Let's go further. The next pathway is going to be the EGFR pathway. This paper was published in June of 2019. Ivermectin reverses the drug resistance in cancer cells through EGFR, ERK, AKT, NF-kappa B pathway. It says here, mechanistically, ivermectin reversed the resistance mainly by reducing the expression of P-glycoprotein via inhibiting the epidermal growth factor receptor, EGFR, not by directly inhibiting P-glycoprotein activity. Ivermectin bound to the extracellular domain of EGFR, which inhibited the activation of EGFR and its downstream signaling cascade, ERK, AKT, NF-kappa B. The inhibition of transcriptional factor NF-kappa B led to the reduced P-glycoprotein transcription. Thus, ivermectin, an FDA-approved anti-parasitic medication drug, could potentially be used in combination with chemotherapeutic agents to treat cancer, and in particular, drug-resistant cancers. So, as you're seeing, ivermectin has multiple benefits, multiple pathways that it's affecting, and I'm going to show you that in graphical format very shortly. I want to say that if you have noticed a trend when looking at these repurposed medications, whether it be mebendazole, whether it be ivermectin, and a variety of other medications, they are using it to restore sensitivity to the standard of care medications, whether it be chemotherapy, whether it be radiotherapy, whether it be immunotherapy, whether it be a monoclonal antibody therapy, et cetera, et cetera, et cetera. What these drugs cannot do on their own, they're being helped by these basically non-chemotherapeutic medications to restore sensitivity to these therapies. And in most of these papers, they talk about using these repurposed medications as a way to then give us a chance to use chemo again. I think that's the wrong strategy. And I think it's going to be further highlighted by the coming papers. This paper was titled "The Important Alpha Beta Specific Inhibitor Ivermectin Affects HIF-Dependent Hypoxia Response Pathways." And so even though our first paper we looked at a variety of antiparasitics having an effect on hypoxia-inducible factors, I wanted to highlight that ivermectin specifically has effects on hypoxia-inducible factors. Hypoxia-inducible transcription factors, HIFs, regulate hundreds of genes involved in cellular adaptation to reduce oxygen availability. HIFs consist of an O2 labile alpha subunit, primarily HIF1 alpha and 2 alpha, and a constitutive HIF1 beta subunit. In normoxic conditions or normal oxygen conditions, the HIF1 alpha subunit is hydroxylated by members of these PHD proteins 1 through 3, resulting in recognition by von Hippel-Lindau protein ubiquination and proteasomal degradation. So basically what happens is when, and I have videos on hypoxia factors with graphics, but the bottom line is when there's normal oxygen conditions or under normal conditions with normal oxygen, the HIF1 alpha and beta are essentially degraded by a proteasome So they're always in circulation, basically, in the case there is hypoxia, but when there's normal oxygen conditions, they just get continually recycled and broken down. When there's hypoxia, that process of breaking it down is stopped and it's activated by hypoxia, mostly through reactive oxygen species. In contrast, reduced oxygen availability inhibits PhD activity, resulting in hypoxia-neutral factor 1 alpha stabilization and nuclear accumulation. Nuclear import of HIF1 alpha mainly depends on classical nuclear localization signals and involves important alpha beta heterodimers. Recently, a specific inhibitor of nuclear import has been identified that inhibits important alpha 1 dependent import with no effects on a range of other nuclear transport pathways involving members of important protein family. In this study, we evaluated the physiological activity of important alpha beta inhibitor ivermectin in the hypoxia response pathway. Treatment with ivermectin decreases the binding activity of HIF1 alpha to the important alpha beta heterodimer. Moreover, HIF1 alpha nuclear localization, nuclear HIF1 alpha protein levels, HIF targeted gene expressions, as well as HIF transcriptional activity are reduced upon ivermectin treatment. For the first time, we demonstrate the effect of a specific important alpha beta inhibition on the hypoxic response on the molecular level. So this is pretty critical because we're going to see here, and this is a graphic that I had used ages ago in my hypoxia-neutral factor series, but you see these little black and red lightning bulbs. And these black lightning bulbs are where hypoxia-neutral factor 1 alpha is upregulating or activating, and a red lightning bolt is where hypoxia 2 alpha is activating or regulating. And we see here that hypoxia 1 is upregulating the glucose transporters, the lactate transporters, all of glycolysis, lactate dehydrogenase A, and then hypoxia 2 is upregulating glutamine transporters, glutaminase, glutamine dehydrogenase, as well as the internal workings of how glutamine is fermented into succinic acid. And so it also has an effect on fatty acid synthesis. So therefore, you could potentially say that the entire Warburg metabolism and the enhanced glutaminolysis and the phenotype metabolism of cancer is controlled by these hypoxia-neutral factors. And when we have, such a strong inhibitor against hypoxia-neutral factor 1 alpha and 2 alpha, such as ivermectin, that could be a potential game changer in the way that the entire Warburg effect is effectively collapsed. Now, let's take a look at the entire kind of landscape of how the Warburg metabolism is established. And there are a variety of ways. So we have arms of this. We have here's hypoxia factor. It's its own thing, but it's also related to the PI3K, AKT, mTOR pathway, as well as c-MYC. So then we talked about the yes-associated protein 1, YAP. That's also related to c-MYC. Then we have the beta-catenin WNT pathway. That's also affecting glucose metabolism. We have a MAP kinase pathway that's affecting glucose metabolism. And then we have the NF-kappa B, or the inflammatory cascade that can activate glucose metabolism. What I would say is that a lot of these signaling cascades, whether it be mTOR, whether it be NF-kappa B, et cetera, a lot of them work through either hypoxia factor 1 alpha or c-MYC. And we'll also see that c-MYC or MYC and KRAS also have an effect at upregulating glutamine transporters, glutamine enzymes such as GLS, as well as other enzymes related to the metabolism of glutamine. Again, it's not just HIF1 in a vacuum. There's a whole host of signaling cascades that are at play here, but HIF1 happens to be one of the main ones, as well as MYC. But what I'm going to show you is going to be pretty impressive here. So I basically took this graphic looking at osteosarcoma model and because it's a nice graphic because it has all of the kind of major signaling cascades that are flowing into showing what it's really doing. It's altering glucose metabolism so that it can survive. And it should also say glutamine metabolism, but it doesn't. So what I did is I looked at all the papers that I could find on a variety of signaling cascades that ivermectin has an effect on. You're going to see that it has an effect on literally all of them, on the beta-catenin WNT pathways, the yes-associated protein, which has an effect on c-MYC, the AKT mTOR pathway, hypoxia factor by itself, like alone, as well as its effects being activated by mTOR and NF-kappa B. NF-kappa B, as well as you'll see later in the coming videos, it has direct effects on glucose and glutamine metabolism as well at the level of the actual enzymes, not just these major signaling cascades. But this just goes to show how powerful potentially ivermectin could be. And that's what a lot of these papers, the next couple of papers, are actually showing. So the multi-targeted drug ivermectin from an anti-parasitic agent to a repurposed cancer drug, it says drug repositioning is a highly studied alternative strategy to discover and develop anti-cancer drugs. This drug development approach identifies new indications for existing compounds. Ivermectin belongs to a group of avermectins, a series of 16-membered macrocyclic lactone compounds discovered in 1967 and FDA approved for human use in 1987. It has been used by millions of people around the world, exhibiting a wide margin of clinical safety. In this review, we summarize the in vitro and in vivo evidences demonstrating that ivermectin exerts anti-tumor effects in different tumor types of cancer. Ivermectin interacts with several targets, including multi-drug resistance proteins, the AKT-mTOR pathways, the WNT-TCF pathways, the purinergic receptors, PAC1 protein, cancer-related epigenetic deregulators such as SIN3A and SIN3B, RNA helicase, chloride channel receptors, and preferentially target cancer stem cell-like population. Importantly, the in vitro and in vivo antitumor activities of ivermectin are achieved at concentrations that can be clinically reachable based on the human pharmacokinetic studies done in healthy and parasitized patients. Thus, existing information on ivermectin could allow its rapid move into clinical trials for cancer patients, and it darn well should. So this graphic is pretty darn busy, and it's a little bit difficult to see because it doesn't really highlight a different color, but effectively you see ivermectin acting all over the cell, looking for ways to shut down cancer. And I think that this graphic is slightly better. And this is a paper that was published in January 2020 saying progress in understanding the molecular mechanisms underlying the antitumor effects of ivermectin. In this review, we summarize the published studies to define the current progress and the characterization of ivermectin. Ivermectin causes cell death in cancer cell lines by inducing PAK1 mediated cytostatic autophagy, caspase-dependent apoptosis, and immunogenic cell death, ICD, through the modulation of some pathways, including WNT, T cell factor, Hippo, and AKT-mTOR pathways. Ivermectin can also affect the growth and proliferation of cancer cells and plays several different roles, such as functioning as an RNA helicase, a small molecule mimetic of the surface-induced dissociation peptide, and an activator of chloride channel receptors, and an inducer of mitochondrial dysfunction and oxidative stress. In addition, ivermectin induces the multi-drug resistance protein and has potent anti-mitotic activity, targets angiogenesis, and inhibits cancer-like stem cells. And I particularly like this graphic because it actually shows, in red where ivermectin is playing roles here. So we have ivermectin in this WNT pathway. You have ivermectin in PPAR cleavage and caspase and apoptosis, the MAP kinase pathway, which was not shown in a previous paper, AKT-mTOR pathway. We see that it has multiple effects on YAP and multiple genes related to cancer stem cells. And this is just, if you want me to be very honest, this is just scratching the surface, because in the next video, we're going to look at cancer's effects on glucose and glutamine metabolism, as well as its ability to induce oxidative stress and actually cause apoptosis within cancer cells through a mitochondrial mechanism. So again, there is a lot to cover with ivermectin and cancer. And there is actually quite a bit published about ivermectin and cancer, which is why it's so frustrating for me to see you labeled as a conspiracy theorist or as a pusher of misinformation when there is so much published literature and really a very bullish stance by the research community, it seems like, at least in the papers that I've read, about its potential. Again, no one's calling it a magic bullet. No one's calling it a cure. No one's using these words. But it is a tool, just like many other tools we've looked at over the year, in having a strategy, an overall strategy of a combination of very key things that block very key things that cancer rely on and that normal cells don't. And I think that we're just getting started with this and it's pretty exciting. So if you like videos like this, please like, share, and subscribe. And until next time.